Hi, welcome to the Bioinformatics Chat. My guest today is Xiang Ji. Xiang is an assistant professor at Tulane University, and we will be talking about phylogenetics today. Xiang, welcome to the podcast.
Hi, nice to be here.
So, Xiang, what is phylogenetics, and why do we care to build phylogenetic trees?
So the fundamental question we want to do in phylogenetics is to reconstruct this phylogenetic tree named phylogeny, and you can consider that as the tree structure that describes the evolutionary relationship between several species. Right? Say you have human, you have mouse, you have dogs, and you can build a tree that say how many million years ago they were one species. So all of this come from the origin of life. So from Darwin, actually.
Right. So in this tree, the leaves would be the species or the genomes that we observe, right? And the inner nodes, the intermediate nodes, would be some distant ancestors that we don't necessarily know what they were.
Yes and no. So we do have some information, say fossils sometimes. And it is quite important, especially when we reconstruct the tree, if we have past information. So that really helps us to say, for example, dating, right? To estimate the divergence time between those species. Say if you have fossils, then you can use the carbon to give you more information outside of our statistical model to say about approximate what the range is about this species, right? And another application of phylogenetics these days is in the region of viral evolution. Right? So there, the leaf nodes will be your sequences from infected individuals. That will be the viral sequences. And in this case, right, first of all, the sampling date of those sequences will tell you some signal of the time, right? And if you think of the tree now, the tree tells you more about how this virus spread over time and over geographic distances.
And so you mentioned quite a few things that can inform us, that can be inputs to the inference process when we're searching for the correct phylogenetic tree. So you mentioned maybe some fossils or maybe the dates where we collected the sequences, but for, so to say, mainstream phylogenetic inference algorithms, the inputs would be just sequences or maybe genotypes, right, that you collect from samples.
Yeah, I think you're right. And to be honest, I think the mainstream phylogenetics inferences we are talking about is probably what people are most familiar with. But the current situation in phylogenetics is we not only consider sequence information, but also consider other traits information. And the trait information can be your the collection location, the longitudinal and latitude positions of the samples, right? And the trait can be other biological traits that you are interested in. That can be the litter size, can be the height, and those things. And but I agree with you. For the tree part, right? Mostly the information of the tree, how they evolve, comes from the sequence, comes from the genetic information, and deep down that is because we assume neutral evolution. Most of the regions of the sequence or of the genome, right? The evolution happen. Most of them are synonymous and they don't, right? It's just the neutral school.
And so when we consider the tree itself, there are certain characteristics of the tree that we care about, right? So we care about the topology of the tree, the shape of the tree. So which species are placed closer together, and we care about the branch lengths. So how much time has passed between the different nodes and leaves in the tree, and also perhaps we care about where the root of the tree is, which is sometimes not obvious, right? Anything else that we care about in a tree?
Yes, I think you know a lot. So yeah, so just to add to your comments, right? So first of all, when we speak trees, there are two kinds of trees in phylogenetics. One kind of tree is called unrooted trees, where you don't really have a time signal. That is mostly because the statistic models we use are time reversible, meaning there is no signal to tell whether where the time flows. Can be either way, and that gives you the same probability. Okay, so for those unrooted trees, right, the unit of the branch length is the expected number of changes per side. So you can see that the unit of the branch length has nothing to do with time, right? And this type of tree is the unrooted trees, and they are the most common trees, to be honest. And the other type of tree is time tree, where you put real time in the branch length. And in this case, right, the unit of the branch length will be the unit of your time. So you say years or million years. And you can see it differs from the previous type, right? The previous type, your branch length has a unit of expected number of changes, right? And so to distinguish them, or say to extract the time information, what we need is another model to describe how fast or slow your gene mutates or your gene sequence change. And that will be the model for your molecular clock. So that will be something I want to add. And so some quantities, right, we are interested in, say, what is the time of the root on the tree, right? That only applies to the time tree, where you also need a molecular clock model to help you date this divergent time.
And so what are some of the methods that are used to reconstruct this tree? And from what we just talked, we need to reconstruct the branch length, we need to reconstruct the topology, and maybe we need to find the root of the tree. So how do people do that?
Yeah, it's a very good question. So first of all, there are, my understanding is there are two major fields or two major applications. First one, if you only need to know one, probably this is the one. That is the parsimony criterion. The idea is you want to reconstruct a tree that has the minimal changes along the tree between the sequences, right? So think of a tree in your mind. Let's say you have three sequences, from one from human, one from dog, and one from monkey, right? And then on this tree you have two internal nodes. And on the internal node from human to monkey, let's assume dog is the farthest, right? Then the idea is you want to reconstruct a sequence at this internal node that has the minimal change towards human and towards monkey. And similarly, you want to have a sequence at the root node that leads to minimum change to the leaf nodes. Right, and this type of criteria that you only want the minimum number of changes is your objective. And that is a way to choose between all kinds of relationships that can describe the number of species you have. And that is the parsimony criterion. And so people found out, right, and so the criterion after parsimony and the criterion most popular these days are called likelihood-based approaches, right? The idea is you use statistic models to describe how the sequences change or evolve along this tree. And from your statistic model, you can build up a likelihood, right? So the likelihood will be the probability of observing your data given your tree and your parameter. And you can have this likelihood, then you draw inference based on your statistics model. That can be another two branches. One is maximum likelihood, another one is the Bayesian approach, right? But so you can see that for maximum likelihood, right, we have this likelihood, and the idea is we want to search through the parameter space, and the parameter includes your topology, and the topology can have your branch length, and your also there are parameters from your substitution process. The parameters describe the substitution along the tree, right? How they evolve, and all those parameters, your final estimate will be the set of parameters that maximize your likelihood, your probability. For Bayesian it's different from maximum likelihood criteria because in Bayesian what you want to sample from the posterior distribution. So that means you're sampling the parameters from their posterior distribution, not only those parameters in the process, in the continuous time Markov chain, right, that you use to describe the substitution, but you also want to sample from the space of the tree topologies. So that will be a major difference. So maximum likelihood will give you a point estimate, and Bayesian gives you a distribution.
And these both approaches that are quite popular today, the maximum likelihood and the Bayesian approach, Bayesian inference, they both rely on the statistical model that you mentioned, or the likelihood, right? How do we calculate, or before we calculate, how do we define the probability of seeing our data given our tree?
So again, think of your three taxa tree. You have sequences from human, monkey, and dog, right? And on this three taxa tree, you have two internal nodes. That would be the most recent common ancestor of human and monkey, right? And then there is a root node. And on this tree, we only observe the sequences on the leaf nodes that say we only know what sequence we have for human, what sequence we have for monkey, and what sequence we have for dog. But we don't know any sequence information in the internal node because we don't observe them. And the idea is, right, so now we have a model, and that model has parameters. And the idea is if for any sequence that we place onto the internal node, right, for any such a set of sequences, let's say we put a sequence for the most recent common ancestor of human and monkey, right, and we have another sequence for the root node. Then given our model and this set of sequence, right, we can describe how the sequence evolve from the root node to the every leaf node, and that gives us a probability. Okay, and this type of probability will be joint because we jointly consider the leaf node and the internal node, right? But that is not the final probability we want because in reality, we don't observe the internal node, and we want to marginalize over. That is the statistic jargon. And it is just equivalent, say, we want to sum over all possible sequences in the internal node that we don't observe, right? We want to sum over all those sequences, so to generate the sum of the joint probabilities to get the final probability, and that is the probability we define. So that's what we say, the probability observing the data given the model. So when we say this, we marginalize, we sum over all possible sequence in the internal node already.
So if we were to implement this algorithm directly, naively, what we would essentially do is for each internal node, including the root of the tree, we would enumerate all possible sequences, right? So if we have n sides, that would be 4 to the power of n sequences. For each sequence, we would calculate this joint probability.
Yeah.
And then we would sum all these individual probabilities, which individually will be very small, but once you sum them up, and then you'll find which set of parameters give you the maximum probability or the maximum likelihood. But of course, this is not a practical solution for anything but the smallest trees and the smallest number of sides. So how do we calculate that likelihood in practice?
Yeah, so in practice, we use Feldenson's pruning algorithm. And the pruning algorithm, the idea is you go from the leaf nodes to and move towards your root node, right? And the name Feldenson's pruning algorithm comes from the developer, right? That is Joe Feldenson. So for the pruning algorithm, right, they work from the leaf node towards the root, and for, let's say, again, using our 3 taxa example, right? So in this case, we want to start from the human and the monkey 2 taxa cherry, right? And then you move to the first internal node, and the idea is now on this internal node, you want to calculate the probability of observing the data at or below this node. Let's imagine the root node is on the top. Okay, so root node on the top, right? And the data at or below this internal node will be the sequences we observe on human and on monkey, right? So that is something we want to calculate. That is the probability of observing all the data at or below this internal node, given the site at certain sequence state. So we are using a nucleotide substitution model as an example. Say that means for every site, we have 4 possible states that will be A or C or G or T, right? So in this case, right, for one site, then you want to calculate 4 probabilities. That will be the probability of the data on this site, right, at human and monkey, given the site is A, or given the site is a G, or given the site is a C, or given the site is a T. So you see that we can calculate this probability, right? And then after having this probability, we can move again towards the root node. On this 3 taxa tree, then we just move to the root node. Then there we calculate the probability of the data at or below this node, given this position is A or C or G or T, right? For the root node, the data at or below the root node is the set of data we have, right? So at this point, right, you have probability of the data given the root node is at A or C or G or T, right? And what you need is the probability of ACGT, right? Their distribution at the root node. And there are usually 2 ways, right? The most common one is we assume your model is at stationary, so you just use the stationary distribution over A, C, G, or T at the root of your substitution process for the root node. So that gives you the probability A, C, G, and T. And you multiply them to get the joint, and you sum over A, C, G, and T, because that's the 4 possibilities, again, to sum over the joint to get the marginal, right? So there you see now at the root node, when we sum over those joint probability, we just recover the probability of the data given the model, right? And easier way to understand is the pruning algorithm, right? So think of the marginal. When we go from the marginal probability to the joint probability, we need to sum over all those possible combinations, right? And so now you see we have a big sum at the outside, and we have something in the inside to sum over. That is the joint probability, where you have all those internal sequences, right? To get this joint probability, we in the internal, right, we have multiple multiplications. So the naive way will be sum over multiplications, right? And then the pruning algorithm is just you sum and multiply at the same time.
So you maybe rearrange, right, the sums and the multiplications so that you sort of like in high school you factor, right, the expressions into like multipliers.
Right, that is the idea. And more technical jargon is the pruning algorithm is just a special case, or is a case of the for dynamic programming algorithm, but it is for the tree structure.
And so yet another way to think about it, why this algorithm is possible, is because the probability of any subtree in our big tree, it shouldn't depend on anything that happened before that subtree, right? So if you condition on the sequence at the root of the subtree, then the probability of that subtree is basically determined, and you can pre-calculate it, right, for every possible value at the root of that subtree. And so you can reuse that computation for all the possible combinations of sequences up the tree or in neighboring subtrees.
Yeah, that is the idea. And it is a very good point, because I think in many smart maximum likelihood calculation packages, right, maximum likelihood packages, they actually do this. They store the probability at the subtree, and when they search through the topologies, right, when they find the subtree, they just reuse it. So it's a very good point.
And so this algorithm to calculate the likelihood, it's been around for quite a while.
The whole field starts from the algorithm. Without the algorithm, it's not feasible.
But knowing the likelihood itself is not quite enough to be able to optimize it, right? It's nice also to know the gradient of the likelihood, which is basically if you change the length of the branches of the tree, how does the likelihood changes? And if you know that, then you can change it in the direction where you increase the likelihood, right? So how do we calculate? How do we modify this scheme to calculate the gradient? And that's basically the subject of your paper, which we'll talk about a bit later. But I'm now curious, like, how did people do this previously?
I see. So we know that the field has been there for decades. We just came up with this algorithm. So for sure, you can do all the things you want without our contribution. So back to the topic, how do you get the algorithm? How do you get the gradient, right? So the pruning algorithm is an analytic form of the likelihood, right? And in the pruning algorithm, you can just normally derive your gradient based on the pruning algorithm, right? So we didn't go into the detail about the mathematical expression, but just think of the way we calculate based on the pruning algorithm, right? We travel through the tree from the leaf node to the root. Let's say you want to get the derivative of the likelihood with respect to one quantity of one branch, right? And all you need to do is to replace that quantity in your travel on the tree in the pruning algorithm by its corresponding analytic derivative form in the pruning algorithm, right? So you can see that you have the pruning algorithm, that is your analytic form of the likelihood, and you can get the analytic gradient based on the pruning algorithm. And the point to note is if you do this, right, then that means for every time you need a derivative, right, you still travel the same direction as the pruning algorithm, as if you're doing the pruning algorithm again. But instead, this time you're doing the pruning algorithm to get the derivative. So that will be something, that is something people do. And so that is the analytic gradient, right? So in the age of using computers, right, we always can get the numeric gradient. That will be whatever your favorite numeric approach is, right? Like if you like the central difference, the finite difference approach, you just do a perturbation around the parameter value and do the central calculation. For an algorithm, right, the first thing we care is the accuracy, and you get the best accuracy from your analytic form. That is done. But the efficiency wise, right? So again, from in our three taxa tree example, right? We have three taxa and we have 4 branches. Let's say we want to get the gradient with respect to each of the branch lengths of the 4 branches, right? And from using the pruning-based algorithm, right, that means we need to travel this tree 4 times. Every time we get one derivative, right? And for a small tree, right, for our tiny little toy tree, that is fine. But when you work with large trees that has thousands of tree leaves, that you don't want to do this thousands of times. So that will be a problem. And that's what we say not very efficient.
Right. So the idea is that you can apply the same pruning algorithm to one particular partial derivative. So the derivative of the likelihood with respect to any particular branch length, right? But that would require this full tree traversal, right? So it would be O of n, right? Meaning it would take a time proportional to the size of the tree. And since you have the number of branches and branch length proportional to the size of the tree, then in total it would be a quadratic algorithm, right?
Yeah.
And that also reminds me of the forward mode automatic differentiation, right? That's what you would get if you were to apply the automatic differentiation in the forward mode. You would also get this one derivative at a time. But I'm wondering if anyone used the reverse mode automatic differentiation on calculating the gradient. So I think it would still be a linear algorithm.
Yeah, so that's a good point. I think that is some, so from what I know, that is some ongoing research in Eric Mason's group at Fred Hutch in Seattle, right? So they are exploring automatic differentiation for the gradient based on TensorFlow. So that they are using TensorFlow's implementation to evaluate and benchmarking the differences. Yeah, so that is one. I agree with you.
But it's interesting to me that it hasn't been done yet because reverse mode automatic differentiation has been around for quite some years now. But I'm curious if you actually looked at the source code for the popular phylogenetic inference algorithms, and do they use a n-squared complexity algorithm, or do they use automatic differentiation? Do they use something else?
I only looked at several popular ones, right? And I don't think they use that fancy algorithm to get if they use numeric gradient. But I think most of them use analytic gradient algorithm for use the analytic form of the gradient evaluations. That is mostly because in the likelihood calculation we have this. When we get the probability transition matrix, we need to do matrix exponentiation evaluations. That is where it's quite sensitive. Let's say there are, I don't remember the exact number, but say 19 dubious ways of calculating matrix exponentials. And I think many popular, like the popular way to calculate is you just diagonalize first, right? You calculate the eigen decomposition of the matrix, and then you calculate the matrix exponential, right? Because of this eigen decomposition, if you just do, let's say, a numeric or finite difference or whatever perturbation, that eigen decomposition could potentially magnify this subtle difference in your numerical error, and it can give you huge trouble. But instead, if you calculate based on the analytic form, right, then you don't have any trouble. It just you can just reuse your probability transition matrices.
If I understand correctly what you're saying that they in effect use this n-squared algorithm, right? So they calculate each partial derivative by doing a full pruning algorithm?
Yes and no. So the yes part is they usually occasionally use this n-squared because we all know, right, it is too expensive. It is quadratic. If you only use it to optimize small number of parameters, right, and that means you're still using ON for each one of them, but the number of total parameters doesn't scale as the number of the tips, right? In this situation, could be that you just want to use it for the several parameters that characterize how A changed to C, how the substitution process inside the substitution process, right? In this case, your total evaluation is still linear because you just limit your number of evaluation, your number of traversals on the tree. And that is plausible, so make it feasible, right?
But speaking about the branch length, you still need to find all the branch length, and there are of the order of n of them.
Right. So that's why I want to separate the branch length from other parameters first, right? So we can still do it on the other parameters, but for branch length, it's mostly because of the heavy duty computation, the quadratic computation, right? You cannot get a gradient for all of them unless you want to pay the quadratic price. And instead, what people do is they do the Newton-Raphson optimization, where you just every time you calculate the derivative and its quadratic derivative, right, on one branch, and you optimize that single branch.
Mm-hmm.
That is another approach. That is, say, you just enumerate over all the branches, right, until you don't see any improvement.
Mm-hmm. So meaning you don't optimize all the parameters at the same time, but you optimize each of them separately.
Right. If you only need to evaluate, that's the same idea, right? You see here, you are still limiting the number of parameters at a time because when you look at only one branch, you are looking at one parameter. So one evaluation is still just one traversal that is still ON, it's still linear.
But once you sum it all up, wouldn't that be quadratic because you have n branches, and optimizing each branch means computing even if it's one derivative, but it's still the linear time traversal, so it still ends up to be n times n.
When you do this type of optimization, I don't think you can stop visiting one branch just one time, even though you optimize them one at a time, but you probably need to iterate over several times.
Right. But I'm saying at
still.
least, right? It will be at least quadratic because optimizing a single branch
Right.
Will still require a linear traversal, an of and traversal. And so once you do that for each branch, however you do that, either you do it jointly by optimizing all the length simultaneously, or you optimize it one at a time, it still ends up to be n times n.
Right. So yeah, we are reaching a different aspect. That is the total time you use in your optimization, let's say. So that's not only the time you spend for one optimization step, but that you need to multiply the number of steps you use, right? And to be fair, we haven't done that comparison yet. And I don't think there is a winner situation in this comparison. That is because usually when you do Newton-Raphson optimization, because you have not only the first derivative but also the second derivative, right? The Newton-Raphson usually converge much faster than you use, say, let's say just only the first derivative. And so you can see that now your comparison, when you compare the total time, you need to think of the time spent in one step and the number of steps. And usually in the Newton-Raphson scheme, the total number of steps is not that many. And when we do, say, gradient based, right, you may just approximate your Hessian information based on your gradient, let's say the BFGS algorithm, right? If you, let's say, you use your gradient over all of the branches, right? You get the gradient of all the derivatives of each branch at a time. And still, that is one step in your optimization, right? And the total time still depends on how many steps you have. And that sometimes can be tricky. If you stare at all those optimizations, let's say, right, the most popular one or the gold standard is still the BFGS. You can see it's optimized. It has very quick jumps at the beginning, right? And at the end, probably you move to 6-7 digits after the decimal point. But every step you increase just a little bit, but you're still running the algorithm. So that will be something different.
That makes sense. But still, what you are suggesting to us is quite compelling. So what you're saying is that you can now, like, you found an algorithm to numerically compute the, or even not necessarily numerically. It's in a way, it's a mathematical expression. There's nothing like approximate about it, so I wouldn't even call it numerical.
Yeah, so our algorithm is analytic expression.
Yeah, so you have this expression for the full gradient that can be computed in a linear time, which is the same asymptotic time that is used to compute the likelihood itself. Within the same time, you can compute the full gradient without doing, gradient, if we're talking about the tree of size n, there will be like approximately n elements of the gradient. Those would be the partial derivatives with respect to the branch length.
2 and -2 for the branch length.
Yeah, in addition to possibly other parameters that you may have, right?
Right.
And as I said, I think reverse mode automatic differentiation would give you the same result, but certainly your version is much simpler to implement. It doesn't require any fancy machinery or TensorFlow. It doesn't require numerical differentiation of matrix exponentiation or anything like that.
No.
How did you come up with this algorithm? How did you know that this algorithm was at all possible? Like, what inspired you to look for it?
Oh, so there is no miracle. And so we know that the pruning algorithm, Phylogenetics pruning algorithm, is just a dynamic programming algorithm, right? So our algorithm again is still a dynamic programming algorithm, right? And the right, when we introduce the way we calculate the likelihood, we say we sum over all possible sequences in the internal node, right? So there what we didn't say is this type of model, because we don't observe the sequences at the internal node, is called hidden Markov model. The sequences at the internal node are the hidden states of your Markov model, right? And when we sum over them, we are just summing over the hidden states of your hidden Markov model, right? So for the hidden Markov model, the algorithm for calculating gradient is well known, and that is the forward-backward algorithm, right? And now you can build up the connection, forward-backward, right? So the forward mode for the hidden Markov model is the thousand-sense pruning algorithm. All right, so the forward mode maps to the thousand-sense pruning algorithm, and you can see that we can employ the backward mode to get the gradient. And all we do is we just extend this backward mode onto the framework of phylogenetics. So how do you do this on a phylogenetic tree? And that will be the algorithm.
Right. So normally these hidden Markov models, they are linear, right? You have a single
Right.
strand of evolution of the hidden state, whereas in phylogenetics you have this recursively dividing states where each unobserved state gives rise to two unobserved states. But you're saying that overall this is a similar situation, yet somehow, as we said, the field has been around for quite some time, and it seems like for the first time someone noticed the connection, right?
Yeah, we were surprised too. People have been using the forward-backward mode for a long time in phylogenetics, right? Let's say when we calculate the posterior marginal distribution, let's say that is used to get the reconstruct the ancestral sequences after you have your likelihood, right? When we do that, what people usually do is they do the forward-backward mode, right? But it hasn't been applied to calculate the gradient, and that's something we just filled in.
Right. And once you have these analytic expressions, there are some other interesting things that you can do. So, for example, you show how to calculate the second derivatives, not all second derivatives, but sort of the main diagonal of the Hessian matrix, which it basically means we differentiate two times with respect to the same branch length, right? And can you talk about what you use it for? What can you do having the extra information, have the second derivative?
Right. So one thing is, let's say if you only extract one of them, and then now you can redo the Newton-Raphson step we mentioned, right? So the idea is, if we just look at the Hessian matrix, right, by its size, it is n squared. Right? If we just fill in the n squared entries, that would be a quadratic calculation already. And if we need a linear calculation for each of them, let's just move to triple, right? So that is a compromise. Say we don't want to pay the price of getting the analytic of the full Hessian, but still we want to enjoy the benefit of knowing something of the Hessian matrix, right? The assumption here is we are assuming that the diagonal of Hessian will tell us the majority of the information on each parameter direction. So that will be the scale of each of the parameters. So that if we know this information, then we can use it. Again, think of the Newton-Raphson scheme, right? In the Newton-Raphson scheme, if we have the full Hessian matrix, right, you can still do the Newton-Raphson with the full Hessian. But instead, if we don't have the full Hessian, but we just want to use the diagonal of the full of the Hessian that is analytically calculated accurate, right? We want to use some information from the diagonal to help us speed up our gradient-based inference procedure. That could be, in the paper we use it to precondition the Hamiltonian Monte Carlo sampler for the Bayesian approach.
And let's say I'm willing to pay the price for the full Hessian calculation. Do you think it would be possible with your approach? Because it seems like the calculations would be a bit more tricky than just differentiating two times with respect to the same branch length.
That's a good question. So first of all, the diagonal. Another reason why we put the diagonal of Hessian in there is if we go through our derivation, you can see the diagonal of the Hessian. All you need to do, right, if you just want to calculate the derivative with respect to branch length, you only need to substitute the Q matrix with the Q squared matrix in our algorithm. So we wanted to show that there is almost no difference calculating the gradient and the diagonal entries of your Hessian matrix. For the off-diagonal parts of the Hessian matrix, you can still take advantage that you have this forward-backward two traversals already, right? And you can take advantage of all those cached values in your intermediate calculations, but you need to pay the price beforehand, meaning you need to derive the analytic expression of the off-diagonal terms. And it is doable. We just didn't put it in this manuscript.
So one thing you experimented with in your paper was so-called rate heterogeneity. Can you talk about what that is and why it's useful to incorporate in your phylogenetic inference?
Right. So rate heterogeneity. So we are now talking about evolutionary rate. Evolutionary rates come from the molecular clock you're using. So again, now we hear the word, right? The word molecular clock. So you see that we are talking about time trees now. What we care is we care about the time at which the species diverge. We care about the timing of the internal nodes, and we want to draw inference on them, right? So you can see that time. So the usual way of saying is time and rate are confounded in phylogenetics. That is because if you look at their likelihood function, right, only the product of time of rate on a branch is the branch length. Is likelihood identifiable, but still, you still can break the symmetry by introducing more or introducing differences between times, right? That is one way you improve your inference. Let's say you bring in more information in the time part. That will be your fossil information or your sampling date, if the sampling dates are different and they are different enough to give you signals, right? And the other component in the time and rate will be the rate part, right? And for the rate, especially for large samples, right, you don't expect all of the sequences on the tree, all of the branches, they evolve at the same time. That is almost impossible, right? And to incorporate the differences, right, how the evolutionary rate change along the tree, you want to model them, you want to put them into your model, right? So that is your modeling choice of the evolutionary rate, how the rate change along the tree, right? To demonstrate our algorithm, the model we choose is an expansion over the very popular uncorrelated rate model. It's more general. It's the more general form of the uncorrelated relaxed clock model, where you have one you have one rate for each of the internal nodes. So each branch has its own rate. But not only that, we also divide this rate to come from a mixed effect model, right? Oh, I think in the paper that is a random effects where we have a grand mean and a random effect for the small residual parts. And you can see now you have one rate for each of the branch. And all we do is to apply the analytic gradient algorithm to get the derivative, first derivative for each of the rate in just linear time in 2 passes, in 2 traversals. And that is the contribution or novelty of the paper.
And you did quite a few numerical or data-driven experiments, so to say, in the paper. Were there any interesting findings, any conclusions you'd like to share?
Yeah. So again, if we just care about the accuracy of gradient, then forget about our algorithm because you can do that with analytic form based on pruning algorithm. The algorithm we proposed helps you to get the same quantity, the same analytic form faster. So the experiments we perform is just to see how fast we can get. So we did experiments in 2 scenarios. One is on the maximum likelihood framework, and the other is a Bayesian framework. In the maximum likelihood framework, we just optimize over the space of the evolutionary rates. And again, we choose the branch-specific rate as an example to use here. So there we plugged in our gradient into the gold standard BFGS algorithm. That is the optimizer we choose, right? And we compare it to the same optimizer, but instead using a finite difference numeric algorithm for the gradient to compare the speed difference. And there we observe a 126 to 234 fold difference. So the fold change is quite coherent with the size of your data. So the size of the data, the larger the size because of the save in your algorithm in the gradient part, right? You change from quadratic to linear, the more time you save. So that is the maximum likelihood part. For the Bayesian part, right? Because now we have the linear time gradient evaluations. That means the gradient evaluations is no longer a computational bottleneck for us. So we can enjoy the rich development over the past decade on gradient-based samplers in the Bayesian framework. And the gradient-based sampler we choose in this case is the Hamiltonian Monte Carlo sampler. And there, so again, Bayesian approach, right? Instead of generating a point estimate, we care about the distribution. And for characterizing your distribution, you use the effective sample size. That is because of the correlation nature of your Markov chain. Because every time you move to the next step from your previous step, so they are correlated. Okay, and the effective sample size is a measure over how good your approximation of the distribution is. And a rule of thumb is you want to maximize this effective sample size as much as possible. Okay, and the measure we use to characterize how good the Hamiltonian Monte Carlo sampler perform over the traditional random walk samplers is we look at among all the dimensions of the branch-specific rate, we get all of their effective sample size, right? And then we look at the minimum, the median, right, to see, because at the end of the day, the only dimension that ruins your Markov chain is the minimum effective sample size. you want the minimum effective sample size to be above certain threshold, so that your approximation for all of the parameters are good enough. So if we just look at the minimum effective sample size, right, and we compare the effective sample size you get per unit time of different samplers, then the Hamiltonian Monte Carlo sampler give us a 16 to 33-fold increase over the traditional random walk samplers. That right, the difference is from the Hamiltonian Monte Carlo because it used the gradient over all dimensions. So that's the HMC Hamiltonian Monte Carlo. It proposed new values for all of the dimensions at a time, compared to the traditional random walk parameter sampler. Right, it proposed a new value for a parameter at a time. So that is the major difference.
Cool. Anything else you want to talk about? Anything we haven't covered?
Oh, we have implemented the algorithm, the gradient algorithm, in the Beagle package, and Beagle package is open source and available on GitHub. Another thing is the Hamiltonian Monte Carlo algorithm, or the sampler we just mentioned, and also the molecular clock model, they are implemented in the BEAST software. So BEAST software, spelled as BEAST, and that stands for Bayesian Evolutionary Analysis by Sampling Trees. So it's another open source software, and if you're interested in Bayesian analysis on evolution on phylogenetics, please take a look over the software. It's one of the most popular software in our field.
Yeah, very, very cool work. I think it's a great contribution to the field of phylogenetics, what you did. And thanks for being on the podcast.
Thank you for inviting me.